Warning. Paclitaxel is a prescription-only medicine for intravenous infusion. All parts of the yew tree are extremely toxic. Any attempt at self-medication with plant material is life-threatening. Diagnosis and treatment of cancer must be carried out solely by qualified medical professionals. Good day, dear friends. Today we will talk about a plant that kills cancer. The yew is not only a graceful evergreen adorning northern forests, it is also the unexpected source of one of the most powerful modern anti-cancer agents. In 1962, the U.S. National Cancer Institute launched a massive screening of thousands of plants, searching for substances able to halt tumor growth. Bark from the Pacific yew, Taxus brevifolia, collected in Oregon, stunned researchers. Its extract dramatically suppressed tumors in laboratory mice. By 1971, chemists Monroe Wall and Munsuk Wani had isolated the active diterpene alkaloid and named it Taxol. The road ahead was long. Of the thousands of promising samples, only a handful reached clinical trials, yet Taxol passed every hurdle. In late 1980s Phase 2 studies it already produced more than 30% objective responses in patients with refractory ovarian cancer, and in December 1992 the drug, Paclitaxel, won full approval. One course of treatment, however, consumed the bark of two mature T. brevifolia trees, threatening an entire ecosystem. Under pressure from ecologists and lawmakers, the manufacturer invested millions in a semi-synthetic route using leaves of the more common European yew, T. baccata. Thus begins our chronicle, a detective story about science, ethics and the struggle for life. Early cell culture experiments showed a peculiar effect. Once Taxol was added, the mitotic spindle froze. We now know the key lies in binding the drug to beta-tubulin. Normally microtubules are highly dynamic, but Paclitaxel locks them in a stable state, blocks depolymerization and derails mitosis. Such pinpoint action overcomes the drug resistance of many tumors, yet it also causes peripheral neuropathy, because microtubule dynamics are critical for long neuronal axons. Chemically, Paclitaxel is a complex structure with 11 chiral centers, multi-layered oxygenated rings and a distinctive side chain. The first total synthesis in 1994 required more than 40 steps and yielded only milligrams of product. On its scaffold scientists later built docetaxel and cabazitaxel, analogues that evade cellular efflux pumps. Beyond freezing mitosis, Paclitaxel triggers caspase cascades, generates reactive oxygen species and modulates PI3K/AKT and MAPK signaling, turning the cytoskeletal dance into a still image. In 1992 Paclitaxel entered standard therapy for ovarian cancer, and soon for breast cancer, non-small cell lung cancer and Kaposi's sarcoma. Meta-analyses from the early 2000s showed that adding Paclitaxel to carboplatin raises 3-year survival in serous ovarian carcinoma by roughly 15%. Today the TC, Paclitaxel plus carboplatin, and TCH, Paclitaxel plus carboplatin plus trastuzumab, regimens are gold standards. In 2025 a combination study of Paclitaxel with the PLK1 inhibitor onvansertib in triple-negative breast cancer reported an encouraging 40% partial response rate. Parallel trials explore pairings with immunotherapies and PRP inhibitors, as well as a chemo plus oncolytic virus strategy that heightens tumor antigen presentation. Approval of the albumin-bound analog Abraxane eliminated the toxic solvent cremophor EL, lowered hypersensitivity risk and increased intratumoral exposure, broadening the drug's reach even further. At first Paclitaxel required large-scale felling of T. brevifolia, but a semi-synthetic route from T. baccata foliage soon became crucial. Yew cell cultures yielded only trace amounts until bioengineers applied CRISPR editing. In 2024 the MPXSN method lifted baccatin 3 titers 12-fold. Discovery of the photo-1 enzyme and a complete 19-step biosynthetic cascade allowed researchers in 2025 to transplant production into Saccharomyces cerevisiae, now generating baccatin 3 at roughly 180 mg per liter. Meanwhile, fast-growing yew cultivars with double the taxane content lessen ecological pressure on wild stands. Once biofactories reach 1 gram per liter, the cost of a treatment course will drop 5-fold. Hybrid reactors, yeast plus microalgae, are already being tested in the EU, coupling drug manufacture with CO2 capture. Moving synthesis from the forest to cleanroom plants marks a breakthrough for both ecology and pharmaceutical safety. The classic solvent cremaphore EL provoked anaphylactoid reactions, so nanotechnology stepped in. Abraxane, albumin particles of about 130 nanometers, use the spark protein to home in on tumors and double penetration into hypoxic zones. Researchers are now designing lipid cubosomes, polymeric micelles, and smart, suprananostructures carrying photoactivators. The capsule dissolves in the tumor's acidic milieu, releasing Paclitaxel plus a radical-generating dye. Clinical pipelines feature combinations with oncolytic viruses and PD-1/PD-L1 antibodies that, alongside Paclitaxel, amplify immune assault. Exosomal couriers bearing Paclitaxel and endogenous antigens already achieve over 50% combined responses in metastatic melanoma. Organ-on-a-chip systems place the final letting scientists watch nanocapsules cross vascular and membrane barriers in real time. The guiding philosophy, less but smarter, defines the horizon. Low-cost yeast biofactories and intelligent nanocapsules are turning Paclitaxel into a precision scalpel against cancer. Ahead lie dose optimization, personalized regimens and, perhaps, a new global oncology standard.